So my name is Brian Meyer. I'm an application scientist with the company. Thank you for joining me today to talk about photosynthesis and the CI340 handheld photosynthesis system. Also joining us from CID Bioscience is Andrea Melnichenko. Andrea is also an application scientist with the company, and she will join us for the discussion and question and answer session at the end of the webinar. So today we'll talk, we'll split the webinar into three parts. The first part we'll talk about CO2 gas exchange and photosynthesis, touch base on who is interested in photosynthesis measurements, discuss some of the features of the CI340 photosynthesis system, and then talk about traditional versus non-traditional uses. Part two of the webinar will actually take up the majority of the time. And this is where we'll do a review of some current research, as well as hear from a researcher who is currently using the CI340 handheld photosynthesis system. The last part of the webinar, I'll introduce the travel grant opportunity, and then we will take questions. Photosynthesis is a critical component of the global carbon cycle, and it is responsible for providing energy to many of Earth's organisms. Because of the importance of photosynthesis and because rates and strategies vary across plant types, the ability to accurately measure photosynthesis is increasingly important. This webinar will present and review current research involving the CI340 handheld photosynthesis system, highlighting unique and innovative uses in the measurement of leaf level dynamics. How do we use carbon dioxide gas exchange to measure photosynthesis? The equation for photosynthesis is shown here. Gas exchange measurements provide direct measure of the net rate of photosynthetic carbon assimilation. Carbon dioxide exchange systems use enclosure methods, where the leaf is closed in a transparent chamber. The rate of carbon dioxide fixed by the leaf enclosed is determined by measuring the change in carbon dioxide concentration of the air flowing across the chamber. We measure carbon dioxide assimilation in a single leaf in order to investigate whole plant characteristics. So here we see a very familiar formula for photosynthesis, and we're going to measure the carbon dioxide assimilation with the handheld photosynthesis system. And again, photosynthesis is extremely important and heavily studied, as about 50% of the Earth is covered in photosynthetic plant life. The handheld photosynthesis system features several different types of sensors. The primary sensor is the infrared CO2 gas analyzer, or the IRGA. There's also an H2O, or relative humidity sensor, a PAR sensor for photosynthetically active radiation or incident light, and three different temperature sensors for leaf, air, and internal temperature near the IRGA. The multiple sensors gather many types of data which can be applied for more than just photosynthesis readings. Direct measurements are taken for CO2, H2O, PAR, incident light, and for temperature. Calculated data is shown in real time on the display and saved with the data. Calculated data includes net photosynthesis, transpiration, and stomatal conductance. The soil respiration chamber, which is pictured on the right side of the screen, uses the IRGA sensor and reports the respiration values as a negative net photosynthesis number, as it measures the release of carbon dioxide from the soil. There's a wide selection of leaf chambers to fit a large variety of leaf types, sizes, and stages of maturity. Closed system chambers are pictured on top of the CI340, and open system chambers are near the front of the analyzer and underneath it. The two chambers here in the front of the 340 with the orange seals on the side are quite adaptable, able to be used with conifers or sedums. They can work as either closed or open system chambers and can also accommodate a stem entering the chamber while still sealing around it. And that's what that orange side seal will do for you. So open systems are configured so that air is continuously passed through the leaf chamber to maintain the CO2 in at a stable concentration. And measurements of photosynthesis and transpiration are based on the differences in carbon dioxide and relative humidity in an airstream that is flowing into the leaf chamber compared to the airstream flowing out of it. The rate of carbon dioxide uptake is used to assess the rate of photosynthetic carbon assimilation, while the rate of water loss is used to assess the rate of transpiration. For open systems, this is determined based on leaf area. In closed systems, the chamber is on top of the 340 here. A leaf is enclosed in the chamber and sealed to avoid gas exchange with the atmosphere. The rate at which carbon dioxide and relative humidity concentration changes in the chamber are monitored. This is determined based on leaf area and leaf chamber volume. Complete equations for photosynthesis and transpiration are available in the user manual or online. Also pictured here is the soil respiration chamber. The environment in the leaf chamber can be controlled with separate control module accessories. The carbon dioxide, relative humidity, light intensity, and temperature can be controlled. There's also a separate module for measuring chlorophyll fluorescence for those interested in photosynthetic efficiency. So who wants to measure photosynthesis? Primarily, plant scientists are interested in photosynthesis measurements, more specifically those interested in phenotyping, or carbon cycling dynamics, or maturity and aging. Very often, I see the handheld photosynthesis system used to characterize stress responses or to study the relationship between photosynthesis and water balance. Other scientists and researchers interested in non-traditional uses also use the instrument and measure photosynthesis. There are many non-traditional uses for a handheld photosynthesis system. Other non-photosynthesis uses include anything that releases or absorbs carbon dioxide or water, such as soil and insect respiration. Carbon credits in climate change can be studied by measuring photosynthesis, carbon dioxide, and water exchange. So now we've come to the second part of the webinar, and we'll talk a little bit about some of the current research that has been done using the handheld photosynthesis system. So there are several predecessors of the CI340 photosynthesis system, but all are based on the same single erga design and technology. The hardware was first released in 1995, with many improvements since then. There have been over 50 publications across 16 different countries, all on a wide variety of photosynthesis-related topics. The top 5 countries are listed in this table on the right side of the screen in order of number of publications. A complete list is available on our website. Now for the second part of the webinar, I will introduce several case studies. Most of the content has been provided by the researchers themselves, and I will present their research and findings. For those who are in attendance, I will introduce them and pass over the microphone so that they may speak about their research. All content will be available online afterwards for those that wish to have a more in-depth review. I had the pleasure of meeting Dr. Benoit Truax several years ago in Montreal. Benoit's group is the Eastern Townships Forest Research Trust, and they're involved in poplar research. They use the handheld photosynthesis analyzer, as well as the soil respiration chamber accessory, to study soil respiration rates in mature hybrid poplar plantations in southern Québec, Canada. Also on his team are Julian, Daniel, and France Lambert. So the materials and methods here provided by Dr. Benoit for the soil respiration study had 3 different hybrid poplar plantation sites that he looked at, and at those sites had 27 different plots. At each plot, soil respiration, including root respiration, was measured using the handheld photosynthesis analyzer, and sampling was done in spring, summer, and fall. So respiration was measured by placing the soil respiration chamber on the soil surface for 2 minutes, during which the CO2 production was measured and the rate was expressed. So this was repeated at 5 random sampling points. Soil temperature and air temperature were also recorded using the sensors on the CI340. And therefore, soil respiration rate, soil temperature, and air temperature could be compared for these plots. So down here at the bottom, we can see a couple photographs of one of Dr. Benoit's researchers using the soil respiration chamber. And here we can see a separate soil temperature monitor, but there's also the temperature monitor for leaf and air temperature installed on the 340. So this is a photo of mature hybrid poplar plantation near the region of La Patrie, Québec, Canada, from 2013. Notice the huge amount of leaves on the ground. It's an extremely deep litter layer. It's also the highest altitude experimental hybrid poplar plantation in Benoit's climatic gradient. So the results from Dr. Benoit's soil respiration study show significant time versus site interactions for soil respiration, air, and soil temperature. He found that across the 3 sites, temperature conditions were rarely similar. There was a strong relationship observed between soil temperature and respiration rate. So the higher the temperature, usually the higher the respiration rate, but this was dependent on the site. It seemed to follow the same trend at the Brompton and La Patrie sites, with lower respiration rates in the spring and fall and a peak in the summer. However, respiration was much higher at Melbourne in late May compared to the other 2 sites, and not different from the respiration rate measured in late July. At Melbourne, air and soil temperatures were also higher during respiration measurements in late May. In the images at the bottom, you can see the handheld photosynthesis system. The leaf chamber, it's installed here on the tripod using the tripod mount, and as well an example of some of their field sites. So this is from a different experimental design on the utilization of a riparian multi-species plantation in the agricultural landscape, studying degraded brooks and ditches of the eastern townships of southern Quebec. Here, the student, Ms. Richards, is shown measuring soil respiration in the center of a red oak plot. The other species that soil respiration was measured on include the hybrid poplar, red oak, bur oak, red ash, and white pine. They wanted to compare silviculture treatments arranged in the split plot, so compare black plastic to the control or weed plots. So silviculture is the practice of controlling the establishment, growth, composition, health, and quality of forests to meet diverse needs and values. It also focuses on making sure that the treatments of forest stands are used to preserve and to better their productivity. The results show important differences between the plastic mulch, which had the lowest value, and the control, which had the highest value, after 3 growing seasons. Surprisingly, the highest temperature was found beneath the black plastic mulch, but this was probably because more organic matter was rapidly burned in the first and second growing season, and the measurements were taken during the 3rd growing season. So the leaf image photos here are courtesy of wikipedia.com, and the photo of Ms. Richards is from Dr. Benoit. So Dr. Benoit's group is expecting to publish again soon. This is a publication from 2012 in the Journal of Forest Ecology and Management. Yielded an 8-year-old hybrid poplar plantations on abandoned farmland along climatic and soil fertility gradients. And you can see in the images at the bottom, this is one of my favorites of the variety of size of poplar leaves. So they have multiple leaf chambers for the photosynthesis analyzer to accommodate different leaf sizes and maturity stages. And you can also see that they collected other data besides just using the handheld photosynthesis system. So they did soil cores and did root density studies as well. The next group we'll talk about research comes from Dr. Josep Peñuelas from Spain. He's with the Center for Ecological Research and Forestry Applications, as well as the National Research Council in Spain. And his group has been using a photosynthesis system for a variety of investigations for several years. This group measures photosynthesis and VOCs in relation to climate, biotic, and abiotic stresses. These are mostly classic uses of the instrument, measuring photosynthesis and water relations. So we'll go through a couple of his recent publications from 2013. The first, needle terpene concentrations and emissions of two coexisting subspecies of Scots pine attacked by the pine processionary moth. And here in the lower left corner, you can see an example of what a male pine processionary moth looks like. So the pine processionary moth is one of the most important defoliators in the Mediterranean region, causing large economic losses and ecological effects. The needle terpene concentrations and emissions may play a key role in the defense of pines. The results suggest that lower terpene concentrations and high percentages of monoterpenes were produced by a combination of emission losses and terpene induction in response to herbivore attack. The photosynthesis analyzer was used to gather data concerning net photosynthesis and transpiration in this study. The next study, intensive measurements of gas, water, and energy exchange between vegetation and troposphere during the Montes campaign in a vegetation gradient from short semi-desertic shrublands to tall, wet, temperate forests in the northwest Mediterranean basin. And this picture in the lower right corner is an example of a shrubland you may see in the Mediterranean region. This was a multidisciplinary international field campaign which aimed at measuring energy, water, and especially gas exchange between vegetation and atmosphere in a gradient from short semi-desertic shrublands to tall, wet, temperate forests. The objective of this campaign was to study the differences in gas, water, and energy exchange occurring at different vegetation coverages and biomasses. The results showed the strong land cover specific influence on the emission of BVOCs, biogenic volatile organic compounds, gas, energy, and water exchange, and therefore demonstrate the potential for feedback to atmospheric chemistry and climate. The photosynthesis analyzer was used in this study to gather data concerning gas and water exchange. Dr. Peñuelas' third publication in 2003 using the CI340 handheld photosynthesis system involved physiological and antioxidant responses of Quercus ilex to drought in 2 different seasons. This is a long-term study over 9 years, and it's a manipulation experiment. The aim was to study the effect of soil drought projected for the coming decades, which is an average of 10% soil moisture reduction. They wanted to see how this would affect photosynthetic rates and water relations, as well as look at the antioxidant and anti-stress defense capacity of Quercus ilex, which is a dominant species in Mediterranean forests. The 2 different seasons they studied were spring and summer. Results show that photosynthesis was limited by stomatal closure in summer. However, a decrease in photosynthesis as a consequence of drought was only observed during spring, possibly due to a low pigment concentration and to an insufficient antioxidant protection. The photosynthesis analyzer was used to gather data concerning rates, photosynthetic rates, and water relations. So Dr. Peñuelas' group is using studies of net photosynthesis and VOCs to gather information for climate change research. Net photosynthesis is only one component of many needed to get the big picture about climate change, and we expect many more publications in the future from this group. So our next case study comes from Auburn University in Alabama from Dr. Josep Kloepper, and he's with the Department of Entomology and Plant Pathology. And this this research was done by one of his former PhD students, and she was looking at drought evaluation with the photosynthesis system. So drought conditions were induced to corn plants at the VH stage of growth, and photosynthesis was measured in the uppermost developed leaf of the plant. Photosynthesis rate, transpiration, and stomatal conductance were measured so they could compare plants under drought conditions and plants under normal watering conditions. And here we can see some of the trial corn plants growing in the greenhouse. So this is a very classic use of the handheld photosynthesis system. Here, instead of a tripod, the hard-sided case is used to prop up the photosynthesis system during measurements. We can see the small leaf chamber is used, and one of the upper mature leaves of the corn plant is having photosynthesis measured on it. So the results of this study came from evaluating the corn plants at the V10 stage of growth after 1 week of being exposed to drought conditions. So you can see from the chart here that under normal watering conditions, we had a high net photosynthesis rate, transpiration, and stomatal conductance. Under the drought conditions, however, net photosynthesis, transpiration, and stomatal conductance were all significantly decreased. Our next case study comes from Michael Bellalou at the University of Sherbrooke in Quebec, Canada. And Michael's just beginning his work with the handheld photosynthesis analyzer. But he is studying under Dr. Bill Shipley, who is with the Department of Biology at the University of Sherbrooke. And Dr. Shipley last published in 2010 using the handheld photosynthesis system in a paper titled The Leaf Economic Spectrum and Prediction of Photosynthetic Light Response Curves. And he used the CI340 and the light accessory module to investigate those light response curves. So Michael's objective is to express drought tolerance of forbs with a functional trait approach. He wants to use net photosynthesis to estimate drought tolerance. He's measuring photosynthesis of different species in control treatment and in a drought treatment. He's using the temperature accessory module and the light accessory module to control temperature and light, and comparing stress versus control treatments at various intensities and temperatures. He uses the tripod mount to hold the CI340 in place while taking the measurement. Notice the lamp on top of the leaf chamber and the heating cooling pad that is controlling temperature underneath the chamber. So here's another image where we can see the plant in the leaf in the chamber. We can see a close-up of the lamp accessory as well as the heating and cooling pad from the temperature accessory. And here's what the accessory module pack looks like. So that he's using the front accessory module for temperature with the big fans on it, and the one in the back corner for the light. We are very interested to see the results of Michael's study on drought tolerance of forbs in the coming months as he works on it this summer. Our next case study comes from Setu Monroe at the University of the West Indies. Setu is the primary researcher who's studying under Dr. Kurt McLaren with the Department of Life Sciences. So Setu is looking at regeneration ecology of a moist forest over limestone in Cockpit country, Jamaica. Cockpit country is pockmarked with steep-sided hollows, as much as 120 meters deep in places, and they're separated by conical hills and ridges. I grabbed a couple photos online to give an example of what the terrain in this area of Jamaica is like. Setu's objectives here were to collect and assess data on forest dynamics, as well as assess major trends and relationships between forest dynamics and environmental variables. He also intends to assign tree species to guilds based on plant ecophysiology and dynamics data. So Setu has some challenges for his project. The first one being that the limestone karst topography with loose surface rocks and the very steep slope incline of greater than 45%. He has to backpack to his field sites, which sometimes can be up to a 4-hour hike. He also deals with daily convectional rainfall. Convectional rainfall occurs pretty much daily in the tropics. Whenever the land warms up, it heats the air above it, which causes the air to expand and rise. And then as the air rises, it cools and condenses as rain. Setu has multiple batteries for his photosynthesis analyzer in order to be able to provide a suitable power source for the equipment while he's at his remote field site. So by having those extra batteries, Setu is able to continue measuring at his remote field site without access to a power source. So here's a great picture where we can see the handheld photosynthesis system set up and how steep the terrain is that he is studying on. So this is also one of my favorite pictures. Notice the blue tarp on the left. This is an essential research tool when working in a wet tropical forest. So Setu uses this not only to protect his instruments to sit on when the forest floor is wet, but to also to cover himself and everything else when it rains every afternoon on him. Setu is also using the light module, and he uses this so he can study or manipulate the light intensity of forest floor plants. He notes several advantages for using this handheld photosynthesis system, especially that it is extremely portable and lightweight. Setu also likes that you can perform on-site calibrations in the field, but as with any instrument, it requires extreme care in the forest environment. And we are also looking forward to seeing some results from Setu's research as he continues this summer field season. So here again is a kind of zoomed out picture of the handheld photosynthesis system with the light module attached, in which he is manipulating the light intensity to reflect forest floor changes throughout the day without having to wait for the day to pass. He can come to the site and manipulate the PAR, the incident light, to mimic morning, evening, or peak afternoon. So our next case study comes from Simone Whitecloud, who is with Dartmouth College. She is a PhD candidate in Ecology and Evolutionary Biology. And she's studying low-lying plants at elevation. And she has an innovative custom leaf chamber. So here we can see Simone. We can see her site is certainly at elevation. This white piece on top of the leaf chamber is actually our custom canopy chamber attachment accessory. So she purchased this piece from CID Bioscience, and then her school fitted a chamber for her out of clear materials so that light can come through and photosynthesis will occur. And this allows her to study these really low-lying carpet plants where she's not able to actually measure the photosynthesis rate on a single leaf because they are so small. So this was kind of her way of getting around that. So Simone actually made this leaf chamber the same size as the soil respiration chamber, which a colleague of hers has at Dartmouth College. And so this will allow her in the future, they can do more research using both of these chambers in conjunction. For now, what Simone is doing is, if you look in the back here, we can see her measuring with the handheld photosynthesis analyzer. In this case, she's put a black fleece sack over the leaf chamber, effectively shutting down photosynthesis. So she's able to get the net photosynthesis measurement from the clear chamber, which includes both plant photosynthesis and soil respiration. And then she's able to put the black fleece over the chamber and get just the soil respiration rates. And she can do the math and subtract them to be left with just the net photosynthesis of her low-lying plants. So Simone is hopeful for some publications in the near future and has certainly come up with a very innovative custom chamber to get around a very small leaved plant for study. Great, all right. So our next case study comes from Lilly Glaesser, who is a student at Southern Illinois University. And Lily, I apologize. I believe I spelled your name wrong in the top slide in the title. So Lily's working with Dr. Stephen Eves, who's with the Department of Plant Biology, and she's actually in attendance today. So I'm going to unmute Lily, and we can listen to her share a little bit about her research and what she's been doing with the handheld photosynthesis system. Hi, Lily. Can you hear me?
Hello.
Good. I can hear you too. Okay, so just let me know when you want me to advance the slides.
All right. So my name's Lily, and I'm from Southern Illinois, but my research is actually in Alberta, Canada. And I work on the oil sands, or the tar sands. After they mined there for about 30 years, the mine is filled in, and I work on reclaiming that area. Is my voice echoing, or is it?
It doesn't sound echoing to me.
Okay.
Is it echoing on your end?
A little bit, but that's okay.
Okay.
I think I can fix it. Okay. So with reclaiming these areas, we look at planting vegetation on it. So the 4 species that I look at are Carex squatalis, Scirpus atrosancus, Triglocan meridema, and Betula glanduloquera. And this is a picture of using the device with Carex squatalis. I'll go ahead to the next.
Okay.
So I measure these rates on the plants, and I'm looking to do non-destructive methods. Because we planted all these plants there, we don't want to sample them by taking their leaves away and running nutrients analysis on that. So we look at their physiological parameters, the photosynthetic rate, transpiration, and stomatoconductance. And from that, we derive the water use efficiency and also the radiation use efficiency of these plants. Next slide. So I take measurements of all 4 of these plants on the reclamation site in 4 moisture classes. Perched, inundated, where it's always wet, fluctuating, where sometimes during the growing season the water table is above the soil, sometimes it's below, and then dry sites. And I take those measurements and I compare them to reference sites, or where these plants are naturally occurring in wetlands around Alberta. And I have 5 of those sites that vary in their most recent disturbance. So UCell is a constructed wetland with all of those plants present, and it was constructed 6 years ago, and very similar conditions as the reclaimed site. And then the mineral soil, disturbed peat, and the roadside ditch were all disturbed by a road built through that area. And their characteristics differ in that the mineral soil is drained a lot when it rains. The disturbed peat was actually part of a bog before the road was built in through there. And then the roadside ditch is much closer to the road, and it's constantly receiving more disturbance from runoff every time it rains through the road, or runs off the road and then floods the ditch. And then the last site is a rich fen that's been well established over 1,000 years ago. So all of these plants occur in those sites, and I compare physiologically what's happening in the reference site to the reclaimed sites. Next slide. All right, so this is some of the data that I've gotten, and it's actually really nice to see that most, for Carex squatalis, all the performance in the reclaimed site is very similar to the natural areas, with the exception for Carex squatalis is the mineral soil. Those plants are not performing, or they're not assimilating as much carbon as they are compared to wetter areas in Alberta. And then, so I take all these data points during one growing season, and to make sure I'm getting an accurate assessment of those plants, I take about 30 readings. So each one of those bars is 30 readings with the handheld device and then averaged. Next slide. So overall, these 4 species are actually performing very similarly on the reclaimed wetland to their reference sites, with a few exceptions. Like the Carex qualis has low photosynthesis on mineral soils. Scirpus is performing almost exactly the same across each site. Triglocan seems to really like very, very wet areas, which during the growing season that I measured was U cell, so it had high stomatoconductance and evapotranspiration at U cell. And then Betula had very low performances on dry soil, and that was all across the board with all its physiological responses: the stomatoconductance, evapotranspiration, and the carbon assimilation. That's what I got.
All right. Thank you very much, Lily. I really appreciate you sharing your research with us today. So I'm going to put you back on mute and finish up the webinar. And then if there's any questions specifically for you at the end, I might unmute you, but we'll wait and see. Once again, thank you, Lily. All right. So now we've shown the handheld photosynthesis system as a beneficial and versatile tool for lots of researchers. For more information on how the 340 could be used in research, please contact our application scientists. Now I'm going to talk a little bit about the travel grant, which is our effort to help support new and upcoming research. Please visit our webpage for more information about evaluation instruments and specifications for the 340 and its various sensors. For the travel grant, we're offering that winners of the grant will receive an extended 30-day evaluation of the CI340 handheld photosynthesis system and a $250 travel grant to present their findings at a conference of their choice. So the guidelines are fairly simple. Submit a written proposal about a page long, outline your idea for an experiment using the handheld photosynthesis system. A proposal should highlight how collecting net photosynthesis data enhances your research. Priority will be given to innovative ideas and concepts. Any of the standard leaf chambers or the soil respiration chamber are available for this project. However, the accessory module will not be available for the project, so please try to plan your proposals accordingly. So we're asking that proposals are due in about 2 weeks, May 1st. We're going to pick the top 3 as winners, and we'll notify them on May 15th. We're trying to kind of speed this one along so that if you are a winner, you can kind of get that plan that you'll have the 340 to do the project with and kind of work that into your summer field season. So winners will have the opportunity to use the photosynthesis system, and that evaluation period is intended to allow winners to carry out the proposed project. At the end of the evaluation period, winners should submit a paper summary or a poster and receipts for travel or registration to a plant science conference to claim the $250 travel grant. Winners of travel grants may also have the opportunity to present their findings at upcoming conference workshops with CID application scientists. You can submit your project proposal and information at the website on your page. And after the webinar, a thank you email will be sent out that will also have some information for you. So now I'm going to open up for questions. If you have any questions about pricing, you can contact sales at CID. If you have questions about the use of the 340 experimental design or method, please feel free to contact either support@CID, which I answer that email. You can contact me at bmeyer@CID. And you can also contact Andrea Melinichenko, who is also joining me right now to help with any questions. Hi, Andrea.
Hi there.
And so you can contact her as well. And she's a plant physiologist, so she's this is right up her alley with the photosynthesis analyzer. All right, so let's see if there's any questions at all. And if not, then I'm going to ask Lily a question. All right, so Lily, I'm going to unmute you, and could you tell me how long it took you to learn how to use the photosynthesis system? Was it a difficult process? Did someone have to teach you?
It was not very difficult at all. It took about a day, not even, just a few hours of learning the controls. It's pretty intuitive. Somebody showed me some of the basic stuff, and then when I was using it in the field, I actually referred to the manual a lot, and it was pretty easy to figure out.
Good. Well, that's what I like to hear. And if you ever have questions, I'm also always here for you too, Lily. If you're accessible to email or the phone, which doesn't always happen when we're out at field sites. All right. Well, I'm not seeing any other questions coming in at this time, so I'll give it just a few more minutes. If you come up with any questions, feel free to type them into the chat dialog box. And if I don't get any more, we'll probably end in about a couple minutes. Lily, once again, thank you for sharing today.
Thank you.
Okay, so I'm not getting any questions that I haven't answered, so we're going to wrap up for the day. The webinar will be available on our website. There'll be a link within 4 or 5 business days, so you can follow that link and review the webinar, the audio, and the presentation. So once again, thank you, Lily, for sharing, and I hope that everyone has a very enjoyable evening.